All right, welcome to your third and final video for the Tuesday, January 7th lecture that you missed. So this is lecture 2B about Mendel and looking at single gene inheritance. We'll be looking at multiple genes later on. So once again, we won't have a chance to enter your clicker answers in this time around because it's a video, but I do highly encourage you to pause the video whenever I tell you to pause and really think through the clicker questions and commit to an answer. You can write down your answer, A, B, C, or D, or E, before you continue with the video, because whether you get it right or wrong, you'll learn a lot by actually doing the problem solving along the way. So please do not watch this video all the way through without stopping. Okay, stop for each clicker question. Alright, so for this lecture, what we're going to do is we'll start with a little bit of background about Mendel, the P's that he worked with and why he was able to succeed where others failed. Then we'll go through some of his single gene experiments in order for you to understand kind of what he did, the calculations that he did, basically to understand the basics of inheritance. And once you know the basics, we're going to immediately apply them to real-life genetics problems, so the kind of work that a genetic counselor would do, so you can start applying them to more difficult and interesting situations. All right? So that's kind of the more advanced stuff we'll be doing near the end of this lecture. So, first thing, let's talk a little bit about what we as a scientific community knew before Mendel did his work, all right? So, even before Mendel did his work, we knew that offspring would resemble their parents, all right? We know that because we've been using that simple fact to breed plants and animals for the past 10,000 years, actually. For example, corn, wild corn, comes in all different kinds of sizes and shapes and colors, even more so than this. But by selectively breeding, say, the yellow seeds from these and growing those up and breeding those together again and again, we're able to kind of exclude some of the darker colored genes. And over time, right, knowing that the offspring will resemble the parents, we were able to breed them to look like the modern corn you find in the supermarkets today. Same goes for wolves, right? Domesticated dogs were closely related to wild wolves, but we're able to domesticate them by breeding the most friendly parents over and over again, and the offspring became more and more friendly as well. We can use the same breeding methods to make little dogs by breeding the smallest dogs we can find together. We can make big dogs by breeding big parents together. If you get wrinkly parents and breed them, you get even more wrinkly offspring. I don't know what they were going for with this one, but yeah, you can breed that too. Breed weird, skinny, ugly dogs. No offense to anybody who has those. But anyways, yes, we know that offspring resemble parents. Another thing we know is that if you breed two parents that look different, the offspring ends up becoming what seems to be a blend of both parents. For example, Plug has a squished face, Beagle has a longish face, offspring has, somewhat squished face. Right? Coloration-wise, notice that the dark back and tail is here, but more faint. Right? Even the tail, notice, super curled tail, straight tail, sort of curled tail, right? So it seems like offspring are a blend of two different parents as well. We can also see that in humans as well, and that if you have two parents with different colored hair, one light, one dark, offspring have hair that's somewhere in between, eye shape somewhere in between, skin tone somewhere in between as well, all right? But if you look more closely, right, and you do more breeding, you'll notice that these rules don't always apply. Like what we know in quotations doesn't always hold. For example, you can have two parents that have brown colored eyes with an offspring that has blue colored eyes, right? You can also have two parents who are little people who have kind of genetic dwarfism, and these three kids all don't have genetic dwarfism, right? This guy's just short because he's young. Also, this is my friend Leroy and Lisa, right? And this is their first child, Kobe, right? Notice blonde hair, blue eye, white skin, but looks exactly like dad, right? And we were really interested because they had another kid recently, so this kid's all grown up now. And this is Tyler, and again, blonde hair, blue eyes, right? So yeah, definitely we're not following the same rules of offspring resembling a blend of both parents, all right? So now looking still at what we knew before Mendel, what did we not know before Mendel did his work? What did Mendel himself not know when he was doing his work between 1882 and 1887? Well, he didn't know that cells had chromosomes, right? That was discovered quite a bit later. He also didn't know that DNA was genetic material. That was way past his time, that we actually did these experiments in order to find if the DNA carried what we inherit from our parents. We also didn't know the structure of DNA as well. That was discovered way after Mendel's time. So how the heck did Mendel succeed in figuring out how inheritance works when we didn't even know what DNA was? Well, there's three major reasons why he succeeded. One is his scientific background and how he approached this problem. The second was his choice of organisms and the traits he chose to study. And the third is he took a lot of efforts to make pure breeding strains before he did experiments. All right, so let's talk about each of these briefly. So first, in terms of Mendel's scientific background and his approach to studying inheritance. Mendel himself became a friar, mainly because he wanted to get a free education. What he wanted to do was teach science, but unfortunately he couldn't pass his oral exams, and therefore he wasn't allowed to go into teaching. But still, he really wanted to contribute to science, which is why he went back to the monastery and he decided to study inheritance, right? How traits get passed on from parent to offspring. Now, he had some kind of lucky advantages. For example, the head of his department, Nestler, just happened to study inheritance in plants and animals. So his background among his instructors was already in inheritance. Nestler studied inheritance in sheep. Another thing that's helpful was that Mendel, during his studies, learned physics from Doppler. Right? Those of you who watched The Big Bang Theory might remember in Halloween, Mr. Bazinga, I forgot his name. Can't believe I forgot his name. But anyways, he dressed up as the Doppler effect, right? So that's the sound. That's what changes a single tone from the horn of a car as it goes by to like, that's the Doppler effect. Anyways, he learned physics from a very good physicist named Doppler. And back then, physics was a much more quantitative and evidence-based study than biology. Biology was much more about just studying and being in awe of the natural world, right? Whereas physics did a lot more measurements and calculations, right? So you had a good background in quantitative study. Now, another thing that was a happy accident in a way was that he went back to the monastery and told his bishop he wanted to study inheritance using mice, but his bishop said he's not allowed to study the mice, maybe because he didn't want Mendel to spend all day watching mice have sex. So essentially he said, okay, plants are fine, breed plants, don't work with mice. So he worked with pea plants instead, which he had growing in the monastery garden. Turns out pea plants are a way better model. So that's what we'll talk about as the second reason why Mendel succeeded, which is his choice of using peas. It wasn't his first choice, but his second choice of using peas, okay, and the traits that he chose within this species. All right, so peas were a great model organism, a really, really good choice for many reasons. One of which is pea plants have a very short generation time, right? So within about 60 days, you can go from a pea seed growing up to a mature plant and breeding and becoming producing fruit and producing the next generation of seeds in just 60 days. And the great thing is, in one plant, you can produce thousands of offspring every generation, right? So you can get lots and lots of replicates. You can also control the mating, right? So if you cut off the male parts of a flower before they mature, then it cannot self-fertilize. Then you can just take pollen from any plant you like and fertilize this plant with exactly, like knowing exactly where the pollen came from. So you can control basically which plant has sex with which plant. Another thing you can also do is just leave the male parts on there, let them mature and let the flowers self-fertilize. Then you know that this plant fertilized itself, all right? Another great thing about the pea plants is that they had very discrete phenotypes, okay, allowing you to kind of study them without any sort of subjective judgment. For example, when you're looking at the pea pods, they're very obviously either green or yellow. Same with the seeds, green or yellow, or wrinkled or smooth. It's very easy to tell the two different phenotypes apart. Also, same with flower color, very clearly, white or purple, right? Even when they're tall and short plants, there's no medium-sized plants. It's either tall or short, okay? So very easy to quantify. So these traits that were chosen were very awesome. One, because as we just went through, there's two discrete phenotypes, which allows for objective results. But another really cool thing is that these traits that can be observed as seeds, they save a lot of time, right? Rather than having to get the next generation to grow up to see if they're tall or short, you can just look at the seeds without even growing them and say, okay, we got this many yellow, this many green, and you're done. Okay, saves a lot of time and space. Also, each trait just happened. Mendel didn't know this to begin with, but each trait happened to be encoded by just one gene that had two different alleles. And for those alleles, one was always fully dominant over the other. Okay, you'll see exceptions to this later on, but for him, his traits always followed these rules. Also, it turns out that for all of the traits that he chose, these genes happened to be located on different chromosomes. All right? Now, it's hard to say whether this was luck, right? It could be that Mendel originally chose some other traits that might have been linked and showed some unusual patterns, and he excluded those on purpose, right? Just so that he would have traits that behaved in a consistent way. All right? But whether it was luck or not, he chose some really good traits to study. All right, finally, another thing that made Mendel's experiments work really well was that he made pure breeding strains first. All right, so he spent 2 years making 14 different varieties of pure breeding strains, so 7 traits, 2 different types each. All right, so 2 things I want you to discuss. One is discuss how you would make a pure breeding purple flower strain. Okay, knowing that purple is fully dominant and the white is recessive, right? How do you make a plant, a group of plants that when they make offspring, all of the offspring are always purple flowered for many, many generations? Okay, what steps would you take to make that? Okay, and the second thing I want you to discuss after that is why is it important that Mendel made these first? Okay, so if you have a friend, if you have a brother at home, go talk to them and see if you can figure this out and think about it. Okay, pause now and write down the best answer you can. Okay, go ahead. Okay, so hopefully you thought about it and kind of in your head or written down step by step figured out how you might make a pure breeding strain. All right, so let's talk about it. So let's say you have a bunch of plants that produce purple flowers, right? But you don't know for sure whether they're pure breeding or not. So what you could do is start by self-fertilizing them. Let them fertilize itself, right? And see what the offspring look like, all right? So if you find that one of these plants makes offspring that are not purple, then you know that this is not a pure breeding flower, not a pure breeding plant, right? So you should exclude that from your population that you're trying to make pure breeding, right? Same with this one here. It produced some white flowers. It's not pure breeding. Let's just get rid of those offspring and parent plant altogether, all right? And then basically go for another generation. And you might find, oh, in this group, we also have a white flower here. So there's some kind of non-pureness here. So let's cut these offspring out. Let's cut its parent out because it's not pure breeding. It's producing some white offspring. And in fact, let's just cut this entire group out entirely because we just don't know where that white allele or where that impurity comes from, right? So if you do this for many, many generations until you have a group that finally never produces white flowered plants, then you're pretty sure that you have a pure breeding plant, okay? So now that you kind of know how he did it, I mean, that's why it took 2 years, because you have to go through many, many generations to be sure that this is pure breeding, a pure breeding group, right? So having done this, having figured that out, so why is it important that Mendel made these first? Well, this is to make sure that this strain is homozygous, right? So from your previous lecture videos, you know that homozygous means that they only have one type of allele, right? So both of their genes have the same allele. And this way, when you take this then and breed it with something else, you know exactly what you're starting with, and your results will be always 100% consistent, all right? If you don't have a pure breeding strain, you wouldn't get consistent results, all right? So in other words, you know what alleles they're carrying, 2 of the same thing, and you can see what happens in the experiment afterwards, all right? So next, let's talk about Mendel's first experiments. So first he started off with pure breeding plants. Of course, he spent 2 years making them. He's got to use them. So what he did was he took pure breeding purple flower plants and he bred them with pure breeding white flower plants. Okay. So when he crossed them together, this is called the P generation. We're taking 2 kind of very different parents and breeding them together. And what he produced were monohybrid offspring. Okay, we call them monohybrid because they are a hybridization of 2 different types of plants, right? Mono because we're only looking at one trait, okay, looking at just flower color. If we're also looking at seed color, then there might be a dihybrid or something like that. We'll talk about those later, okay? So these monohybrid offspring, these offspring that are a mix of purple and white flowered parental plants, turns out they all had purple flowers, okay? This is called the F1 generation, or the first filial generation, okay? So what was, when he did this experiment thousands of times, he always found the same result. He always found that the F1 generation had purple plants. And he didn't just do it with flower color, he also did it with seed shape, seed color, pod shape, pod color, all these kinds of things. And always we found that the F1 generation had just one of the parents' types, and not the other one, and not some sort of blend between the 2. Okay? So if you were Mendel, would you say that these results supported or rejected the blended inheritance hypothesis, that belief that we had before Mendel came along? All right, so pause now and think about which answer you think is correct. Okay, so hopefully you chose B, which is to reject it. I mean, some of you might have said C, that there's not enough evidence to decide, but in fact, the fact that these offspring are not a blend, right? They're not kind of a light purple flower, which is what you'd expect. Blended inheritance hypothesis would say that the offspring are a blend of what the parents have. Since our results consistently point against that, right, we're able to reject the hypothesis. We're able to say that this is not in play here. This is not true in this case. Okay? Notice that it's actually really quite a bit easier to prove that something is untrue than it is to prove that something is true, right? When it comes to hypotheses, right? Well, as an example, let's say that you had a hypothesis that Anderson Silva is the best UFC fighter in the world, all right? And the more times that he defends his title and wins, right, the more evidence you have to support that hypothesis that he is the best fighter, right? If he beats 10 people, he defended it 10 times, then we're 10 times more sure, we're more and more sure that he is the best fighter. But as soon as he loses a fight, which he did by breaking his leg in a horrible way, then that hypothesis is gone. We know for sure already that in a single fight, that Anderson Silva is not the best fighter. Whoever that guy was who beat him is now the best fighter. All right, so notice, that's why I'm always trying to reject hypotheses, because that's a lot easier to do than to prove one. All you can do is find support that something might be true, but it's very easy to reject it. Okay, we talk more about this later on once we talk about experimental design. Okay, so an important thing that Mendel did, right, was that he also did reciprocal crosses. All right, so in one experiment, he might take pollen from a white flower and put it into the female part of a purple flower, and he also tried the other way around, took the pollen from a purple flower and put it onto the female part of a white flower. So basically made the purple and the white flower the mother in both in different experiments, and he found that he always got the same results. All right, that's an important point to keep in mind, right? So far, the traits that we've been studying, the results are not really sex specific. It doesn't matter which one's the mother, because otherwise you might think, oh, okay, the offspring always look like the mother, but no, they always look purple, no matter who the mother is. Okay, so next, let's talk about what happened. Let's try to figure out what happened to this white flower trait, right? Did it get dominated? Did it get deleted? What happened to it? So Mendel wanted to figure that out himself, so he took 2 of these F1 plants and bred them together, or he just took one of them and let them self-fertilize, okay? So it's in this mating between 2 F1 offspring that we produce a monohybrid cross. A lot of people, when they see monohybrid cross, in an exam question, they just automatically think P generation, F1 generation, F2 generation, this whole thing is monohybrid cross. It isn't. Remember, this is a monohybrid. It is a hybrid of these 2 plants for just one trait. So monohybrid cross is a cross between 2 monohybrid plants. All right. So in the F1 generation, when we cross them or self-fertilize them, we're doing a monohybrid cross. Okay. So monohybrid, again, is heterozygote for one gene made by mating 2 pure breeding plants. All right. So when we do a monohybrid cross and look at the offspring, the results of that monohybrid cross, we found, hey, look, we got white flowers again. The white flower phenotype reappeared, okay, in this F2 or second filial generation. So pause now. So think about what this means. Pause now and choose your answer. Okay. So what does this mean? What does it mean that this white flower allele came back? Well, it tells us that all of the above is right, right? So some people looking at this might say, oh, the white flower allele was just weak, it's gone, it never comes back, right? So it wasn't lost in this F1 generation. Also, we didn't somehow blend these 2 types of genetic paint together and lost the white altogether, right? The white flower allele sticks around, right? But also tells us not only did it stick around, when you're making gametes, the white and the purple separate so that it's possible to get 2 white alleles together to make a white flower offspring in this generation. Okay, so we'll explore this a little bit further here. Okay, with Mendel's laws. So one of the Mendel's first laws is the law of segregation. So law of segregation basically says that the alleles that these plants got from each parent, right, they're kept as separate units. So 1 copy of purple, 1 copy of white is kept within this plant, kept separately. Don't blend them, they don't get lost. They're both in existence inside every cell of this plant. Okay. Then when this plant does meiosis to produce pollen or produce ovules, it then separates those parental alleles. Okay, so either just 1 purple or just 1 white allele gets passed on to its offspring. All right, so that's what the law of segregation is it takes what it got from its parents, keeps it separate, and then separates them again to put just 1 of them into the offspring, into the pollen or the ovules, into the gametes. So the gametes will have just the purple or the white from its parents. Okay. So to show this to you in terms of chromosomes and genes, so using the modern science, we know that Mendel didn't know, right? A monohybrid would have 2 different alleles, big A allele for purple, small a allele for white, right? And during meiosis 1, we separate 1 chromosome from the other, so this one has kind of 2 copies of big A, 2 copies of small a, and then in meiosis II they separate individual chromosomes, and we have half of the gametes, half of the pollen, for example, having big A, the purple allele, half of the gametes or pollen or ovules having small a for the white flower allele. Okay, so half of the gametes get big A, half of the gametes get small a, right? So next, let's continue on and explain this whole situation here in terms of modern genetics. Okay, so here's another question. Please pause and think about it. Okay, so continuing on. So the genotypes of the P generation are big A, and small a. They're pure breeding plants. The dominant must be purple, recessive must be white. Okay. So how do you know that both plants are homozygous? All right, so think about that. Pause now. Okay, so coming back, because they're pure breeding. Okay, so we said at the beginning that this is a true breeding or pure breeding purple flower plant. In order to be true breeding, it must have only one allele, so we never have something else appearing. Okay. Next, how do you know that purple is dominant over white? That this is big A and this is small a. Okay, pause now. Okay, fairly straightforward answer is by looking at the offspring, right? When you first did this experiment, you may not know which one's dominant yet, but when you see that the offspring are purple, then you know that the purple parent must have 2 big A's and white parent must have 2 small a's. So, next question. Pause now. Okay, so continuing on. So now looking at the monohybrid cross, right? We know that the monohybrid cross is, well, one hybrid, big and small, crossed with another hybrid, big and small. So looking at the percentage of F2 offspring that should have white flowers, well, the answer is 25%, and there's actually 2 ways to find that answer. Feel free to use whichever method makes the most sense to you. Okay, so one method to figuring out the results, or predicting the results of this cross, is to do a Punnett square. Okay, so Punnett square will help you find out the probability of any of the offspring genotypes. Okay, so the way I usually do this is I make it so that whatever the male parent of this cross is producing in its gametes, we'll put on the side here. So in terms of pollen, right, the male parts of this flower will produce half of the gametes will have half the pollen will have big A, half will have small a, and in terms of the female part of the flower, the ovules, half of those will have big A and half of those will have small a as well. All right, so we've set up the possible gametes from the female parent, the possible gametes from the male parent, and then down here we're going to put what the offspring will have when you take one thing from the male and one thing from the female. So if we get a big A from the male, big A from the female, the offspring will be big A, and therefore be purple, right? Here we have big A, small a, big A, small a. They'll also be purple because big A is dominant. And then finally, one of these 4 will get small a, and be white flowered. Okay, so once we're done the Punnett square, once we filled out what the offspring possible genotypes are and figured out what their phenotypes are, we can then calculate the probability of any of these things happening, right? So we calculate it with a fraction. So what we do is we put the number of kind of possible offspring that have the trait we're looking for. In this case, we're looking for which ones will have white flowers. So this one here will have white flowers. And we divide it by the total number of possible plant types, right? So there's 4 different possible outcomes. So overall, the probability of getting white flowers would be 1 white flower plant out of 4 different possibilities, 4 equally likely possibilities, and that gives us 0.25, or if you wanted it as a percentage, 25%. Okay. Other way of figuring out this probability is by using probability calculations, right? So to get a white F2 offspring, right, we basically have to give it 2 small a alleles. All right, so let's calculate the probability of that happening. So the probability that the pollen from this father plant will contain a small a is 1/2, right? This parent only has big A and small a alleles, so there's a 50-50 chance that this parent will give the small a instead of the big A. Same thing goes for the ovule. There's a 50-50 chance in this parent that that ovule will contain a small a, right? So that's another 1/2. So the probability that you get small a from dad and small a from mom, right, is basically this multiplied by this. So 1/2 times 1/2, which gives us 1/4, which is 0.25 or 25%. Okay, so whatever method you prefer, go ahead and use that. I'll accept both types of work in the exam. Okay. All right. Well, yeah, do make sure you explain your work clearly, though. Either one will get you the right answer, but if we ask you to show the work, make sure you show the work clearly. All right, so now that we know that 1/4 or 25% of these offspring will have small a, and therefore have white flowers, right? Here's some kind of some genetics problems for you to solve. Okay, what's the probability that this purple plant here? Okay, we have the plant; it's grown up already. What's the probability that this purple flowered plant is a carrier for the white flower allele? Okay, so in other words, when we say carrier, as we said in the pre-lecture video, a carrier is a plant that is carrying and hiding a recessive allele. Okay, so what's the probability that this plant is big A, small a? All right, so think about it, choose an answer, and then we can continue. So pause now. Okay, so hopefully you calculated your answer. So the answer might be a little, might actually be wrong. A lot of people get this question wrong, so maybe it's a good thing that I'm not counting these clicker questions. The answer is actually 0.67 or 0.6666. All right. The reason for this, okay, think about it. Of the possible offspring of this cross, right? These are the ones that give you big A, small a. But we have extra information here, right? We know that the plant we're looking at does not have white flowers, right? Therefore, this one here is no longer a possibility, right? This is not one of the possible outcomes out of 4, right? We remove that, and therefore that leaves us only 3 possible outcomes, right? 2 of which are big A, small a. So the probability now that we know that there's no white flowered, this is not a white flowered plant, right? There's 2 big A, small a's out of the 3 possible purple plant outcomes, right? So our answer is 0.67. Okay, here's a little bit more practice. So what's the probability that this plant here, once again, this purple flower plant here, is big A, right? Pause now. Okay, so hopefully this more of you have gotten this one right. So the answer is 0.33, because once again, we know, having already grown up this plant, that this plant does not have white flowers. It cannot be small a, not possible, right? Therefore, only one of the 3 remaining possibilities are big A. Okay, so that gives us, Okay, so that gives us 1/3. Okay, in case you didn't see that, right, here's another question. Probability that the next F2 offspring that's produced will be big A, right? So think about it. We have this cross happening. We're making 1 more seed for the F2 generation, which has not grown up yet. What's the probability that that new seed will grow up to be big A, right? Pause now and think about it. Okay, so hopefully you got 0.25, all right? Because in this case, since we're now producing a seed that hasn't grown up, we have no idea whether it's going to have purple or white flowers. All 4 of these possibilities are equally likely now. Okay, so since we're producing a new offspring, right, that we don't know anything about, we have to consider all 4 possibilities, and of those 4, only 1 will be big A. So 1 out of 4 is 0.25 again. All right, if you have any questions about this, feel free to come by my office hours or the TAs later on. Okay, so notice that when we're looking at the Punnett square, right, we can kind of, and looking at also the results of the actual results in the F2 generations, when we actually look at Mendel's plants and count everything up, we can tell that the phenotypes, in terms of the colors we see, there's 3 purple for every 1 white, right? There's a 3-to-1 phenotypic ratio, okay? 3 purple for every 1 white. Now we can predict, using this Punnett square, that among those 3 purples, like 2 of them would be big A, small a, and 1 of them would be big A. All right, so 1 big A for every 2 big A, small a for every 1 small a. Right? That's what we get from the Punnett square. But we can't actually observe that directly, right? We can't see what the genotypes are just by looking at the plants, right? So how did Mendel prove that this actually works, right? That this phenotypic ratio we see of 3-to-1 is actually 1 of these to 2 of these to 1 of these, right? When the big A and big A, small a plants look exactly the same, we can't tell them apart at all. All right. So let's solve this puzzle together. All right. So to go to another example with using puppies because they're cute and fun. So for Labrador retrievers, right, the black color of fur is dominant over the brown color of fur. Okay, so we'll give the black phenotype a big B allele and the brown phenotype a small b allele. okay? So let's say you find a stray black lab in the street where you know nothing about its parentage, and all you know that it is that it has black fur, all right? How would you figure out Wolfie's genotype? Okay, so pause and think about that. Okay, so as far as how you'd figure out Wolfie's genotype, the easiest and fastest way to do it would be to test cross Wolfie. And this is a specific genetics term, doing a test cross. So a test cross is if you take your kind of dominant phenotype but unknown genotype individual and you mate it with a recessive, homozygous recessive individual, a brown lab, okay? Now, we know that Wolfie is either big B or big B small b, so either homozygous dominant or heterozygous, right? We don't know which one, right? Now, if Wolfie is actually big B, right, and we know that its mate is small b, because otherwise it wouldn't have brown fur, then look at the offspring. All the offspring will have a big B from Wolfie, and therefore have big B. All of these should be black, okay? But if Wolfie's actually heterozygous, big B, then there's a 50-50 chance that its offspring would actually be brown, right? And the other ones would be black. So pretty much, if you see any brown offspring at all, that's pretty much proof that Wolfie is heterozygous, right? Because if he was homozygous dominant, you shouldn't see any brown offspring at all, right? So here's a question for you to consider. Let's say that from this test cross, there were 6 puppies. All of them had black fur, all right? What would you conclude then? Pause and think about that. Okay, so the thing is, you can't really be sure still, right? For all you know, the 7th offspring you might have might be brown, right? Yes, it's somewhat unlikely. Like it's a coin flip, right? It's a 50-50 chance if he's actually heterozygous that you get a black or brown one. So it's kind of like flipping a coin 6 times and getting head 6 times, right? Which is still not that unlikely, right? So the more times you do this test cross, the more puppies you see, the more likely it is that it's this condition and not this. Okay, so let's keep that in mind. All right. So getting back to this pea plant example, right? Using that test cross example from the puppies, we can pretty much apply it to this situation as well, all right? So if we want to know exactly what the genotypes are of these purple plants here to find out if the genotypic ratio is really what Mendel predicted, then you pretty much have to test cross them, okay? And you should test cross them with a white flowered plant because then you can be sure that you're mating with something that has small a. So you would test cross them and see if any of their offspring are white flowered. All right. So here's a bit of a challenge question for you to think about. So in reality, Mendel didn't do a test cross. He didn't take these purple plants and breed them with a white flowered plant. He just let them self-fertilize instead, right? So what's the pros and cons of his approach? Okay. So we already said that test crosses are kind of the go-to way to figure out, say, Wolfie's genotype. So why did he let them self-fertilize instead? What are the pros and cons of that choice? Okay, so pause and think about that. Okay, so in terms of the pros of letting them self-fertilize instead, it's a lot less work, right? To basically do deliberate crosses, you need to cut off the male parts at the right time. You have to use a paintbrush to just transfer the pollen from one plant to another. It's a lot easier just to let them sit and fertilize themselves. The con of this, though, is that if you're taking your unknown plant and letting it mate with itself, and they happen to be heterozygous, right? There's only a 25% chance that you will see the white flower phenotype, right? Whereas if you actually test crossed it with a small a individual properly, there's a 50-50 chance. So there's a much better chance that you'll see this evidence of heterozygosity if you do a test cross versus self-fertilization, right? But the thing is, that's fine because when you're doing crosses with pea plants, you're making so many offspring that even though you have half the chance of seeing it, you'll still see it because you're looking at hundreds of individuals potentially. All right, so his approach actually made sense in that less work, and you'll see the results anyways because you got hundreds of offspring. All right, so moving on, let's do some more practice now. Okay, so if up until now you've been a little bit confused, this is a great opportunity, since this is a video, to go back and rewatch what we just went over to make sure you really understand the basic concepts that Mendel taught you and the clicker questions and thought processes and calculations that we've done so far. Once you understand the basics, let's move on to some real-world genetics problems. Okay, so let's imagine that you're a genetic counselor, someone who works at a hospital, at a counseling center, where you would take the results of genetic testing say for potential parents, and explain, okay, what are the risks for your offspring? All right. So let's say that your client, you have a new client named Jane, and she's really terrified because her mom just recently began suffering from Parkinson's disease, which is a genetic disorder that can be passed on. Right? Now, Jane's parents did some genetic testing and found that Jane's mom is homozygous for the recessive Part2 allele. Okay, so it's a recessive allele. Because she has Parkinson's disease caused by this particular allele, she must have 2 bad copies of it. So she's homozygous for the recessive allele that causes Parkinson's disease. Thankfully, though, her dad's genes are all wild type meaning fully normal. Okay, so assuming that other genetic tests came back normal for Jane's parents, meaning that there's not some other allele that can cause Parkinson's disease as well to confuse this picture, let's say we're only looking at the Part2 gene, right? What can you tell Jane? All right. So pause now, think about it, and commit to an answer. Okay, so the answer in this case is that Jane for herself is actually totally safe. She doesn't have to worry at all because while her mom does have 2 kind of recessive bad copies of this allele, which is why she has the disorder, because her dad has only good copies to give her, and that's the dominant allele, right? Jane for sure has a big B, small b, has a heterozygous genotype, and big B being normal and dominant, Jane's totally going to be fine. All right. Next question. Read this and decide an answer. So pause now. Okay, so coming back now. So now we're not talking about Jane herself getting Parkinson's disease. She's worried about her offspring getting it. So we're not going to get into exactly what the offspring has yet, because that depends on her husband or the sperm donor, what gets contributed from the other side. But just look from Jane's side, what's the possibility that she's going to be passing on her bad allele? 50-50 chance, right? We know for sure she has 1 good copy, 1 bad copy. So half of her eggs will have, unfertilized eggs will have a bad copy, half will have a good copy. Okay, so once again, Jane is heterozygous, half big B, half small b. Okay. All right, next question. So pause, read the question carefully and commit to an answer. And so pause now and do that. Okay, coming back now. So as I said, we have to look at Jane's husband in this case as well to know what he's bringing to the equation. Okay, so in terms of background for Bob, we know that both of his parents are carriers, right? Meaning that they have big B, small b. And he knows for sure that he doesn't have Parkinson's disease. Okay, so he's old enough to have shown some signs if he had it. Okay, so the probability that Bob is a carrier, that's still something we need to know for them to know what's happening with parents, Jane and Bob's potential offspring. Okay, so looking at the Punnett square of Bob's parents producing him. Okay, so we know that Bob's parents are big B, small b, big B, small b. So here are the possible genotypes of any of their offspring, including Bob, right? But we know for sure that Bob does not have Parkinson's disease, so small b is no longer a possibility, right? So among the 3 remaining possibilities, 2 of them would make him a carrier. So the probability of Bob being a carrier is 2 out of 3 possibilities. Okay, that's as accurately as we can predict Bob's genotype right now. Okay, so let's assume that Bob is a carrier for now, just to simplify the equation. So if Bob is a carrier for sure for Parkinson's disease, okay, and we know that Jane is for sure a carrier, what's the probability that their offspring will have Parkinson's disease, okay, and have the actual disorder? Okay, so pause now and think about that. Okay, so let's look at the situation. So if we assume that Bob is big B, small b, is a carrier, and we know that Jane is big B, small b, right? The possibility that their child will have their offspring is 1 out of 4 equally likely possibilities. So one quarter, or 25%, is the probability. Okay. Now here's another question. So we don't know that Bob is a carrier. We don't know that for sure, right? So based on everything we know so far about Bob's parents and Jane's parents and what their possible genotypes are, what is the realistic probability that Bob and Jane's first child will have Parkinson's disease? So this is a harder question. Think about it. Make your best guess at it. Write down your answer. And we'll continue after this. So pause. Okay, we're back. So the answer is 1/6, and I'll explain why. Okay, so in order for the child to have Parkinson's disease, which is what we're calculating the probability of right now, right, both of these first 2 events must occur. So number 1, Bob must be a carrier, because it's not a sure thing. And number 2, Bob and Jane must both pass on their bad alleles to the kid. All right, so the way we calculate this overall probability, right, of this happening and this happening is we multiply them together, right? Remember, for probabilities, if the probability for the first event is 2/3, probability of the second is 1/4, the probability of this and this happening is this times this, okay? So 2/3 times 1/4 will give us 2/12 or 1/6, okay? So notice it's less likely that both of these occur than any one of them occur, which kind of makes sense, okay? So think about this carefully, review it again in the video if you like. If you have questions, feel free to email me or to come talk to us during office hours. Okay, next question. So, completely new person now. So Mark has, let's say, a few extra toes, all right? So this trait is relatively common, actually, and it's caused by a dominant allele this time. So whereas Jane's Parkinson's disease was caused by a recessive allele, now we're talking about a dominant disorder, okay? Mark's dad also has a few extra toes as well, okay? Mark's mom, on the other hand, is wild type. She is normal. She has 10 toes, okay? So knowing only that, what is Mark's genotype? So think about it, pause now. Okay, so those of you who chose B, big T, small t, are correct. Because think about it, Mark's mom, we only know that her phenotype is normal, right? But because to be normal, you must have two recessive normal alleles, right? We already know her genotype for sure, okay? So we know that Mark must have gotten a small t from mom, and even though we don't know his dad's exact genotype, right? His dad could be big T, or big T, small t. We know that Mark must have gotten a big T from his dad, right? He must be big T, small t because otherwise he wouldn't have extra toes, okay? So we know that Mark has the dominant allele from his dad and the recessive allele from his mom because that's, number one, all that his mom can give him. Number two, he must have gotten the big T from somewhere to have extra toes himself, okay? Now, let's look at the next generation now. So Mark is married to Melissa, and we're going to talk a bit about Melissa's parentage to find out what she might be carrying. Okay, so Melissa knows that her dad is homozygous dominant, right? And her mom is heterozygous, okay, for this particular gene. So what's the probability that Melissa is heterozygous? Okay, so kind of testing your terminology here. So think about your answer, pause now. Okay, so this one's in a way slightly easier if you remember your terminology. So Melissa has a 1 in 2 chance that Melissa herself is heterozygous because her dad is homozygous dominant. She must be getting a big T from dad, whereas her mom could give her a big T or a small t, right? So there's kind of a 50-50 chance that her mom will give her a small t and make her heterozygous, all right? So once again, this is Melissa's dad, homozygous dominant. This is Melissa's mom, heterozygous. This is the possible genotypes that Melissa herself can have, right? And half of these are heterozygous, right? 2 out of 4, or 1/2, are heterozygous, right? So using the Punnett square, that's how you'd figure it out based on the information we have from Melissa's parents, okay? Next question. What's the probability that Mark and Melissa will have a child that is wild type, that will have a normal number of toes? So pause now and think about it. Okay, coming back. It's 1/8, and let's work our way through it again. Okay, so in order for Mark and Melissa's child to have small t, and therefore have wild type or normal number of toes, right? Number one, Melissa must be heterozygous, right? She must be carrying at least one small t allele, and we know that the chances of that happening is 1/2, right? And once Melissa is heterozygous, and we know that Mark is heterozygous, right? We still need both of them to be lucky enough to pass on the small t allele to their offspring, right? So Mark is heterozygous. We're assuming that half of the time Melissa will be heterozygous, and even if that's true, there's still only 1/4 chance, 1 in 4 chance that the offspring will be wild type. Okay, so to figure out the probability of wild type child, we need the 1/2 chance that Melissa is actually heterozygous, times it by the 1/4 chance that they will then have a homozygous recessive or wild type offspring. Okay, so that gives us our 1/8. All right, so you'll really need to know these calculations for your assignments, so do make sure that you understand that well. Okay, so at this point, we're pretty much done with the Tuesday stuff. you are now ready to just try your practice assignment, which is now available in Mastering Genetics. So go to Mastering Genetics, click on the practice assignment for week 2, which is this week, and try some of those. Once you have the hang of those, don't do the quiz yet because you may want to see the Thursday lecture first, but you can go ahead and do some practice exam questions as well, which you can download from the Canvas site for this course. It's in the lecture schedule table at the bottom of the homepage. And they're called PEQs, or practice exam questions. Generally speaking, you want to do lots of practice because not only do you want to get good at finding the right answer, you want to be able to do it quickly and accurately as well. Okay, and that takes practice. All right. If you run out of questions, you can always find them online as well. For example, for these types of questions, just look up practice genetics problems or Mendel practice problems or something like that online, and you'll get lots and lots of extra problems. Okay, that's pretty much it for now. So thank you so much for your patience and understanding as baby's pushing his way out. I will see you all on Thursday, hopefully, and see you then. Thank you.